Hello, everyone. First, I want to thank the Dravet Syndrome Foundation and the organizers for the invitation to present our lab's research. Today, I want to talk about metabolic control of Dravet syndrome. As you know, the major mainstay therapies for epilepsy depend on rebalancing the excitation inhibition through ion channels and synapses. An alternate view is to achieve seizure control through metabolic pathways that underlie these circuits and can provide fuel choices to control seizures. Our goal is to uncover novel mechanisms that are metabolic in nature by using old and new technologies and further investigating the structure and function of metabolic organelles. The process of epileptogenesis, as you know, can arise from either acquired insults, such as status epilepticus or trauma, but as also genetic mutations in genetic epilepsies. And we understand in great deal the types of insults, as well as the cellular, as well as circuit processes that underlie the etiology of the acquired epilepsies. However, much less is known about how genetic epileptogenic processes evolve, and we often focus on mechanisms that are very close to the genetic mutation rather than common pathways. In our lab, we are interested in how metabolic alterations can be initiated by genetic mutations that arise with the epilepsies. So two simple questions that we asked in the Dravet syndrome area was whether metabolic dysfunction occurred in a model of Dravet syndrome, and if so, could we target the metabolism to improve outcomes associated with Dravet syndrome? We chose to do this in the Dravet syndrome zebrafish model. And this is, as you have heard from Scott Baraban throughout several years, is a convenient vertebrate neural structure that has great sequence homology and is genetically malleable. So it's very useful for precision medicine purposes and has fairly low cost in maintenance. For our purposes, it's important for its ability to be subjected to high throughput screening, and it is permeable to drugs. So it's a really important screening process to understand alterations in metabolism. So we chose to study the SCN1 LAB mutant of Dravet syndrome, which is a very useful preclinical zebrafish model developed by the Baraban lab. This is a natural mutant in which one of the genes for the sodium channel in the zebrafish has been mutated, and it replicates the symptoms of Dravet syndrome in that there's spontaneous epilepsy, temperature dependence of seizures, resistance to the standard pharmacotherapy that the kids have, and sensitivity to ketogenic substrates. As you can see on the left corner here, there's hyperexcitability that can be uncovered by assessing the field potential recordings. And there's increase in the hyperactivity, which is the seizure-like behaviors that these zebrafish display. So important to remember is that these are larval zebrafish, and they can be easily bred and developed in lab models. So our objectives were twofold. First, we were interested in measuring two important energy producing pathways, which are glycolysis, so glucose metabolism, and mitochondrial metabolism in the zebrafish model. And our strategy was to normalize any alterations that we observed in the zebrafish model. So I'm going to talk a little bit about data generated by a postdoctoral fellow in the lab, Manish Kumar, and then go on to talk a lot about data generated by a current postdoc in the lab, Dr. Raji Banerjee. So we used extracellular flux analysis, which is a method that you can measure two important energy producing pathways, that is glycolysis, which is measured by the efflux of protons into the media, and which makes the media acidic. And you can measure consumption of oxygen, which assesses the mitochondrial respiration. And interestingly, we can place the zebrafish, each larva per well, in a 24-well format. And change the media and subject these larval zebrafish to different substrates and inhibitors in a high or medium throughput manner to assess the extracellular flux. And what we observed in our early studies, you can see there's a little zebrafish embryo in each of these wells. And what we saw on the left here is there was a decrease in basal glycolysis rates. And we also observed a decrease in mitochondrial respiration. So baseline glycolytic rates and mitochondrial respiration rates were observed in the SCN1 LAB or the Dravet syndrome mutants zebrafish model. Moreover, we went on to conduct a pretty thorough mitochondrial energetic assay. And here you can see in this panel A, this is just a cartoon of what we should typically observe in cells that are subjected to inhibitors and activators of mitochondria. And what we observe in the panel B is that the wild type zebrafish have a higher rate overall than the SCN1 LAB. And if you measure the different parameters and quantify them, what we see is a decrease in the maximal respiration rates, proton leak, and a parameter known as the spare respiration capacity, which is the ability to mount a response in respiration when stressed in the SCN1 LAB mutants, which is the Dravet syndrome mutant, compared to wild type controls. However, the non-mitochondrial oxygen consumption, i.e. the oxygen consumed by other enzymes other than the mitochondria, were not altered. So this suggests a general mitochondrial dysfunction in this zebrafish model of Dravet syndrome. Next, we asked whether this could be due to any changes in mitochondrial DNA itself or through the electron transport chain complexes, the activity of these enzymes. And what we observed was there was no alteration in the relative mitochondrial DNA content or enzyme activities, which suggest a flux was altered in mitochondria. Moreover, and interestingly, when you treat these larval DS zebrafish with ketogenic substrates, as you can see here, these yellow dots, you can actually improve their metabolism to the level of the wild type siblings. So this suggests that ketogenic diet elements can improve the respiration in this model. We went on to do a glucose metabolism PCR array, which was very specific to zebrafish genes that control glucose metabolism. And in this experiment, what we found was there were many genes that were altered, but most prominently altered were two important genes that were involved in glucose metabolism, which are phosphoenol pyruvate kinase 1, PCK1, and PCK2. And as you can see on the right here, what these genes do is they control the process of new glucose formation, which is gluconeogenesis. And so gluconeogenesis is the process by which glucose is made from substrates, and it's kind of the opposite of glycolysis. However, gluconeogenesis uses a distinct set of enzymes and steps, and which are not common to glycolysis. So what do we know about glucose and mitochondrial metabolism in the genetic epilepsies? One thing we do know is that glucose hypometabolism or decreased metabolism is observed in many human epilepsies, including in children with DS. In general, during epileptic seizures, in the ictal phases, there's hypermetabolism observed in many different cases, followed by interictal periods of hypometabolism. And because hypometabolism tends to be the majority of the phase, the overall observation in metabolism is a hypometabolism. Secondarily, mitochondrial defects have been observed in muscle biopsies of children with DS, and they may be due to glucose hypometabolism. As we know, glycolytic substrates are what feeds mitochondrial respiration. So one reason for this could be an overall decrease in glucose hypometabolism. And what we also know is that children with Dravet syndrome often respond positively to ketogenic diets. Another idea is from imaging studies in neurodegenerative diseases as well as in epilepsies, temporal lobe epilepsy in humans as well as in models, in animal models. And there is a general observation that there may be some hypometabolism when you look at glucose through imaging techniques. So there is a decrease in these conditions. And this on the right bottom is Parkinson's disease brains, and in the left corner here on the top is Alzheimer's disease brain. So there seems to be a general metabolic dysfunction that is common, not just in neurodegenerative diseases, but also with the temporal lobe and other types of epilepsies. What about larval zebrafish? How can they reflect glucose metabolism? So it's very interesting that in larval zebrafish, the enzymes which were downregulated, which are the phosphoenol pyruvate kinases, PCK1 and 2, are actually important biomarkers for glucose, and they also are changed in their expression or increased in expression to provide glucose during embryogenesis. Therefore, we thought maybe these enzymes which were downregulated were causing a decrease in glucose concentrations in zebrafish. So we went on to measure glucose levels in the zebrafish, the wild type, and the SCN1LAB mutants. And what we found was there was a significant decrease in absolute glucose concentrations in the larval DS zebrafish. Now, one thing to remember is that this was done in the whole embryos. And so we don't know what was happening in the brains versus the periphery. And when we looked at total protein concentrations in these zebrafish, there was no difference, which suggests that there was a specific decrease in glucose concentrations or levels in larval SCN1LAB zebrafish. In the next steps, we asked whether we could upregulate or improve this glucose via gluconeogenesis, which is the two enzymes, and correct all the metabolic defects and perhaps the consequent seizure behaviors. And so the question overall was whether metabolic alterations could be harnessed to improve seizure control in this mutant model. And our strategy was to take a small shelf library of compounds that were known to increase the expression of either PCK1, which resides in the cytosol, or PCK2, which resides in mitochondria, or improve the flux of through the enzyme itself, and ask whether these could improve metabolism in the DS model. And many agents such as serotonergic agent, adrenergic agent, starvation, branch chain amino acids, as well as ketogenic diet substrates are capable of improving these gluconeogenic pathways. So our experimental design was simple. We had 3 steps. The first was validation of known PCK activators. We treated the zebrafish, the mutant larvae for 3 hours with different concentrations of the agents that are known to increase expression of these genes. And then secondarily, we would take these hits from this model and put them through phenotyping assays. And we had a behavior assay and the metabolic assays. And lastly, if there were any hits in this phenotyping assay, we would test them in electrophysiology. So I'm going to go through 3 of these steps here. And the first was using pharmacological agents to upregulate the expression of these gluconeogenesis genes. So 3 hours of treatment with a small shelf library of compounds. And I'm showing you the data for one concentration, which is 10 micromolar of each of these drugs. And what we found, and this is a busy slide on the left, but we found that many of these compounds were able to increase the expression, as we expected from the literature, of PCK1, more so than PCK2. There was only a few that increased the expression of PCK2. And these included many different agents that included this compound PK11195, cis-retinoic acid, fluoxetine, which is Prozac, and many adrenergic agonists. I'd like to point out some selectivity for these compounds between the different enzymes that we assessed. So what we see here are these PCK activators, the known activators that increase PCK1 expression more so than PCK2, but they did not alter the levels or the expression of GCK, which is glucokinase. Moreover, steripental, which is an anti-epileptic drug that's useful in children with Dravet syndrome, did not change the expression of these, any of these enzyme transcript levels. The second phenotypic assay that we subjected the hits from the first assay was the behavior-based phenotyping. So this is the Knowles-Daniel vision system, which is a locomotion behavioral assay system that tracks the zebrafish's locomotion and swim behavior. And we conducted two screens. Screen one was conducted in the University of Colorado labs, and same compounds were then put through another level of screening in Dr. Baraband's lab at UCSF. And what we observe here in the reds, the warmer shades, are the inhibition in a concentration dependent manner. And what we see that these compounds such as PK11195 and some of the others such as fluoxetine were picked up by both screens, as well as steripental, which is our control agent. And of note, we focused on PK11195, and as you can see, in this assay, any kind of velocity that is lower than -40 is a positive hit, and you can see a concentration-dependent inhibition of locomotion or seizure-like behavior, swim behavior, in the by PK1195 in this assay. Our second the phenotypic screen was the metabolism-based phenotyping. In this, we conducted the extracellular flux analysis, which I described earlier, and we subjected these compounds to these metabolic screens. And our goal here was to see if any of them, after a certain number of hours of incubation, was able to improve the baseline glycolytic and or mitochondrial respiration rates. And you can see in this pretty busy graph up here in the top right, the glycolytic rates were improved by this compound PK11195, as well as some of the other compounds as well. And so were the mitochondrial respiration rates. And these were pretty close to the wild type controls that we observed the deficit in our zebrafish mutants. So from there, we asked, what about the glucose concentrations? So here's an assay for glucose levels, and were they able to actually correct the glucose deficit that we saw? And the compounds PK1195 again was able to rescue the deficit in glucose concentration, as well as some of the other compounds. So this data suggested that the behavior assay as well as the metabolic assays were able to pick up some hits through our screening paradigm. So here is a very busy table that has some of the hits from some of the early assays that we conducted. And you can see we tested several of the TSPO ligands, which I'll describe in a second, which the PK11195 belongs to, as well as other compounds, adrenergic and flux activators, as well as steripental. And what you can see in this screen is that this PK11195 was the only compound that actually came up positive in all of the different screens. Now, several of the compounds were able to alter some, if not other types of assays, and we'd not really go into the concentration dependence or the conditions under which these would be working, because we were most interested in understanding if our hit compound was effective in our final assay. So the final assay was confirmation of the anti-seizure property of PK11195, and this was conducted by Scott Baraban in his lab. And what he did was used electrophysiology in larvae that were briefly paralyzed with pancuronium and immobilized, and he conducted an assay for local field potentials. And you can see on the left here is a excitable status in the SCN1 LAB that were treated with control media. And you can see many events that are occurring, and the interictal plus ictal events are on the left here in the dark circles. And when you treated these larvae for 3 hours with this PK11195 at a 10 micromolar concentration, there was a general inhibition in the ictal events, which can be observed in the center here. However, the duration of the events was not altered. And so this really confirms that this treatment of this compound at concentrations that was capable of inhibiting the metabolism also suppressed electrographic seizure activity. So the question is, does this really occur through this PCK pathway? And to test this, we used the wild type zebrafish larvae, and instead of mutating the gene for PCK or knocking it out by CRISPR, what we used is a pharmacological approach. And the reason for this, that we could achieve transient inhibition of the enzyme, and that because this gene is so important for embryogenesis and development, we didn't think it was going to be possible to actually address that issue with gene knockout. So we used this compound 3-mercaptopicolinic acid, and the question was whether it would have any effect in wild type zebrafish that mimicked the Dravet syndrome zebrafish. And what we found was an increase in the swim behavior that resembled the Dravet zebrafish. As you can see here, it looks very similar to what I showed you earlier in the SCN1 LAB mutants. And what was also interesting is that this compound was able to mimic the glucose deficit. So this is a concentration dependence of the compound in the glucose concentration assay in the wild type zebrafish. And we were able to reverse the effects of this using the PK11195. So the glucose concentrations were then normalized by PK11195. So this suggests that this mechanism could be due to its effects on the PCK itself. So the next question you may have is, well, how does this compound, what is the mechanism of action? So we've uncovered a compound through our metabolism-based screen that is effective in controlling this glucose dysregulation and mitochondrial metabolism, as well as inhibiting seizure activity. So what we do know about this compound is it is a ligand for the 18 kilodalton protein known as TSPO, or translocator protein. And that protein is a complex of the peripheral benzodiazepine receptor. And we do not, at the current level of our understanding, really understand exactly what the mechanism is. But TSPO has been associated with many processes such as cholesterol transport, the synthesis of heme and steroids, and it controls the peripheral processes linked to the benzodiazepine receptor, or by indazepines. Most commonly, this PK1195 has been used as a radioligand for PET imaging to really look at neuroinflammation, which is a really interesting area. And the way it works is that it is an antagonist and a partial agonist of this ligand. So we, at this point, do not completely understand its mechanism, and we are trying to do this. But what has been observed by certain studies is that this PK1195 may have some anti-seizure effects in mice as well as zebrafish. And our best guess is that it normalizes glucose, altered glucose levels. So in a diabetes model, which has low glucose, it can actually control the glucose levels and bring them back to normal levels. In this case where we had the low glucose levels, or we could, in diabetes where you have high glucose levels, it brings them back, whereas in this condition where we have the lower glucose levels, it actually brings it back to normal. And so we think it may be a sensor for glucose concentrations. And just to understand the role of the TSPO mRNA itself, the protein expression, we asked whether there was a change in the SCN1LAB mutants. And what we found was that compared to wild type zebrafish, the levels of TSPO messenger RNA are upregulated in the Dravet syndrome model. And if you treat these zebrafish with PK1195 at the same concentrations that we use in our studies, you can actually bring this back to below the vehicle. So it's controlled, the protein itself can be controlled by the ligand that we used, which really suggests that this TSPO expression may reflect an increase in neuroinflammation through microglial activation in the Dravet syndrome model. So in summary, what I've told you is that the Dravet syndrome's mutant zebrafish have spontaneous seizures and metabolic deficits, specifically decrease in the genes for gluconeogenesis. They have metabolic defects in glycolysis as well as mitochondrial respiration, and treatment with one of our hits from our screen showed an improvement in all of these parameters, including the expression of this gluconeogenesis genes, normalizing of the glucose levels, improvement in respiration, and suppression of seizures. So this suggests, to my earlier point, that metabolism-based anti-seizure therapies may be useful in treating the genetic epilepsies, at least in this model of Dravet syndrome. We do not really understand the next steps yet, and we are looking into those. And what we want to do is understand how PK1195 may be serving as a sensor for glucose concentrations. And so with that, I'd like to end my talk. Thank you for your attention. And I want to thank in particular the postdocs who conducted this work, Dr. Manish Kumar and Dr. Raji Banerjee, who did the majority of this work, which was recently published in Brain Communications. Mr. Chris Yoon did some of the bioenergetics assay, and Dr. Shane Rowley helped establish this model. And in the Baraband lab, I'd like to thank all of Scott's lab members, as well as Scott himself, who did the electrophysiology. A special thanks to New R01 that Scott and I received from the Child Health Foundation and the Dravet Syndrome Foundation for funding Dr. Raji Banerjee's postdoctoral fellowship, and our zebrafish facility, and Angie Ribera and Kristen Artinger's labs in our institution for helping us establish zebrafish as a model for Dravet syndrome. Thank you so much.